This video is part of the lecture Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava. Previously, we defined the concepts of landrace and cultivar as crops resulting from mass selection in which individuals share common characteristics, but they are not entirely genetically or phenotypically uniform. In this video, we will see what are pure lines or true breeding lines and how can they be produced and improved with controlled crosses followed by line selection. True breeding lines are comprised of genetically identical individuals that produce genetically identical descent. This is because true breeding lines are homozygous, so when they mate, they only produce homozygote individuals, identical to them. Because there is no genetic heritable variation in the true breeding line, they cannot be improved by mass selection. Instead, true breeding lines can be improved by the insertion of new traits. One way to do this is by controlled crosses followed with line selection. Controlled crosses are simply crosses in which both parents, the pollen donor and the pollen receiver, are known. They can be done manually, taking the pollen of one plant and depositing it on the pistil of another one. Or in the case of wind-pollinated crops, they can be done by simply castrating the pollen receiver plant so that it doesn't self-pollinate, and then letting the wind pollinate it with pollen from another donor line. The first step when doing a controlled cross is to select for traits or genotypes of interest among all the available genetic variation for the breeder. A local farmer can obviously work with the genetic variability they have in the field, but big commercial breeders often use genetic variability stored in seed banks. These facilities store a vast diversity of seeds that include not only true breeding and commercialized lines, but also cultivars and landraces, crops that are no longer used by farmers because they have been replaced by more productive or resistant lines, as well as wild relatives of the cultivated crops. For each stored accession, all available information is carefully gathered and then recorded, starting from the name of the crop and its genetic status, where and when the seeds were harvested, to all the characteristics that might be of an interest for a breeder. A description of the crop phenotype, a measurement of its yield in different environmental conditions, all resistances or susceptibilities to different types of stress and pathogens, and so on. The breeder can use all this information to select the crops which are the most suited for their breeding program. So the crops will not only have traits that they wish to be directly improved, but also other traits that will facilitate the breeding program from the start. For example, if a crop is destined to be sold on the African market, the starting crops from the gene banks would be chosen so that they have as many possible traits that are already adapted to the African climate. Let's see how the breeding process of pure lines goes step by step. We start with two parental pure lines. Each has some desirable characteristics. Let's say that the one on the left has high yields and the one on the right resistance to salinity. We would like to combine these two traits together so that we have a crop that has a high yield and high salinity tolerance. The first step would be to cross these two lines with each other so that we obtain an F1 hybrid that has inherited half of the chromosomes from the productive parent and the other half from the resistant parent. In theory, now we already have a crop that combines the genes for both of these characteristics. However, this crop is a hybrid and not a true breeding line. Indeed, if we let the F1 hybrids to self-pollinate, because of the recombination between the chromosomes inherited from different parents, the F2 generation will be genetically heterogeneous. And although on average all individuals will have inherited some productivity and some resistance traits, there will be huge phenotypic variation among these individuals. So after we have done the initial controlled cross, we need to eliminate all undesired genes from the genome of the new crop so that only the desired traits remain in a homozygous state. And this can be done over the course of several generations of mass selection coupled with self-pollination in the field. At each generation, individuals that have the desired combination of traits are selected and left to self-pollinate, whereas all other individuals are eliminated. This combination of mass selection and self-pollination will result in a quick elimination of the undesired genotypes in the population and also a reduction of the heterozygosity within the individuals. Within 6 to 10 generations of reproduction, a new true breeding line can be obtained that has high productivity, high salinity tolerance, and no undesired traits from its original parents. How does selfing reduce heterozygosity in a population can be learned in another video of this playlist. In some species, self-pollination is not as efficient or sometimes even not possible. And to eliminate the undesired traits, we can instead proceed with back crosses. So after the initial cross between the pure parental lines to produce the F1 hybrid offspring, the F1 offspring is crossed with one of the parents, and here it will be the parent with high productivity. So the next generation, F2, will inherit half of the genes that come from the highly productive parent in the cross, and the other half will be a recombined chromosome from the F1 generation. This process is also repeated for several generations until a uniform true breeding line is obtained that has both desired traits from its original parents. Controlled crosses do not have to be limited to the combination of two distinct pure breeding lines. Carefully designed breeding programs can combine several F1 hybrids together so that multiple traits of interest can be simultaneously inserted in the crop. For instance, a very complex breeding program is the one that produced the IR36 rice line that started with the line IR8, which is semi-dwarf, has high productivity but is very susceptible to diseases. This line was crossed with a Taducan lines, which is another local line from the Philippines that is resistant to fungal blast. So their F1 offspring already has some of the desired characteristics, but it can be further improved. And to do this, we will not only proceed to line selection of the F1 offspring, but we will further combine it with another line that is obtained from the controlled crosses of two other pure breeding lines, the TKM6 and the Taichung native 1. So now we have two F1 offsprings, each that has a combination of desired characteristics that will be crossed together to produce a new hybrid that has high productivity and multiple resistances to different diseases. And again, the breeders don't have to stop here. They have also introduced a third hybrid line resulting from the crosses of two other rice lines that were further back crossed to produce a new line with desired traits. So after several of these crosses and then followed by selection of which eliminates the undesired traits, we end up with the IR36 line that is semi-dwarf, which means it can bear heavier loads without breaking, early maturing with high grain quality and multiple disease and insect resistances. Controlled crosses, when coupled with line selection, are a powerful tool for the plant breeders. They can produce true breeding lines, which will always produce offspring of the same quality as the original crop. Because of their genetic stability and uniformity, true breeding lines can be easily certified for a broad commercial use. This breeding technique is still used today because it is the most efficient way to improve quantitative genetic traits, in which numerous genes need to be inserted at once in order to obtain the desired phenotype. The disadvantage of pure lines is also their genetic uniformity. Because they show no genetic variation, they are not capable to evolve and adapt to climate change in the environment, contrary to genetically diverse land races and to some extent cultivars. This is why, whenever a pure breeding line faces a new pathogen or environmental challenge, the whole breeding process needs to be undertaken anew in order to produce a new tree breeding line that can survive the new environmental conditions.